Okay, our first speaker is Dr. Tom Steeger. He is a Senior Science Advisor in the EPA Office of Pesticide Programs Environmental Fate and Effects Division, where he works as an ecological risk assessor and contributes to developing new tools for assessing ecological risks from conventional pesticides. Tom, feel free to begin.
All right, well, thank you very much for this opportunity to speak here today on EPA's efforts to assess and manage risks of pesticides to bees. Slide 2. During this presentation, I'd like to briefly touch on the declines in pollinator health and in factors associated with those declines. I'd like to talk about the national strategy to promote the health of honeybees and other pollinators, and EPA's pollinator risk assessment process and efforts to mitigate risks from pesticides, and finally on EPA's efforts to expedite the review of products used to control varroa mites, which are a serious pest for honeybees. Slide 3. This upper graph is taken from the National Agricultural Statistics Service, and it's actually pulled from the National Research Council's report on the status of pollinators in North America that was published in 2006. And what it shows is the number of colonies that are managed for honey production in the United States declined from roughly 6 million colonies in the 1940s to about 2.8 million in 2006. The bottom graph depicts the same data plotted from 1970 to 2015, and they're plotted as discrete points. And what it shows is that from 1982 to 1986, there are no points, and that's because NASS, the National Agricultural Statistics Service, was not conducting the survey at that time. And when it resumed the survey, the methodology had changed such that fewer beekeepers are actually captured in the numbers being recorded. Also depicted on the lower graph is when varroa mites were introduced in 1987. It was followed by a decline in the number of managed colonies for honey production from 1990 to 1995. However, from 1996 to present, the numbers of managed colonies for honey production have actually stabilized. The current estimates place the number of managed honeybee colonies in the U.S. at about around 2.88 to 3.18 million. What the graph doesn't depict, though, is the level of effort that beekeepers have to go to maintain those numbers. Slide 4. This is a depiction of data or a graph that was compiled by the Bee Informed Partnership and researchers at the University of Maryland showing overwintering losses of managed colonies. And prior to the introduction of varroa mites, who again is a serious pest to bees, in 1987, average overwintering losses were about 10% in the United States. And after the introduction, overwintering losses averaged about 15%. When CCD, or colony collapse disorder, was first identified in 2006, since that time, the 10-year average for overwintering losses has been about 28%. Based on the most recent survey year, 2016 to 2017, overwintering losses were reported at 21.1% and were actually the lowest since they've been recorded since 2006. Annual total losses, though, still represent about 35%. The gray bar in this graph actually depicts what beekeepers have reported to be economically sustainable losses, and they have averaged over the past 10 years about 15%. More recently, they've risen to 17%, but it's clear with total annual losses at 35% and the average 10-year average overwintering loss is 28%. It's well above what beekeepers consider to be economically sustainable. The 2017-2018 report for overwintering losses should be released in the very near future, and it'd be interesting to see how those numbers compare to previous years. Slide 5. There are multiple factors associated with colony collapse disorder and declines in honeybee health. No single factor has been identified as a cause. Those factors include diseases such as the fungal disease Nosema, parasites such as varroa mite, and small hive beetle. the loss of forage habitat through the conversion to agricultural production or urbanization, and because of decreased foraging habitat, a decrease in the nutritional status of bees. Pesticides are clearly a factor, as are bee management practices in terms of moving bee colonies over long distances. These factors are working in combination with one another. However, there's no exact combination, or the exact combination remains unclear. More recent data developed by researchers at the University of Maryland, working in conjunction with the Bee Informed Partnership, though, suggests that the varroa mite, in combination with particular viruses, such as Varroa destructor virus, can be a particularly virulent combination. Slide 6. In response to declines in honeybee health and other pollinators, the work was initiated on the national strategy. Slide 7. Because of the reports and declines of some pollinator species and continued elevated losses in honeybee colonies in the United States, recognizing that these losses were not limited to the United States alone, in 2014, President Obama issued a directive to federal agencies to increase and to coordinate their efforts to improve bee health by developing an integrated strategy. The memorandum also specifically directed EPA to engage state and tribal agencies in the development of managed pollinator protection plans. Slide 8. EPA and the U.S. Department of Agriculture co-chaired the federal task force, and in 2015, the White House released the national strategy to promote the health of honeybees and other pollinators. This strategy outlined multiple commitments by federal agencies to promote honeybee health. It included a pollinator research action plan to address uncertainties, and it identified a public education plan, and it emphasized the need for public-private partnerships. Slide 9. The overarching goal articulated in national strategies include reducing honeybee overwintering losses to less than 15% within 10 years, restoring monarch butterfly numbers to about 225 million by the year 2020, and over the next 5 years, having 7 million acres of land available for pollinators to forage. Slide 10. EPA made multiple commitments within the national strategy. Those commitments included an effort to assess the effect of pesticides on bees and other pollinators, to restrict the use of bee-toxic pesticides on crops requiring managed or contracted pollination services, and to engage state and tribal partners in the development of managed pollinator protection plans. EPA also committed to expediting the review of registration applications for new products targeting pests such as mites, which again are very harmful to honeybees. Slide 11. With respect to the first goal, EPA has been evaluating risks of pesticides for well over 20 years. Throughout this process of learning about the multiple factors associated with pollinator declines, EPA's focus has been on ensuring that the best science is brought to bear on assessing the potential role that pesticides may be playing in the declines of some species of insect pollinators. Slide 12. Well, before the national strategy was released in 2015, in 2011, EPA issued interim guidance on exposure and effects data on bees that we would be recommending. This guidance was based on the agency's understanding of the evolving risk assessment process that was highlighted in a Society of Environmental Toxicology and Chemistry Global Pelston workshop on pollinator risk assessment that was held in 2011, and also on work that was underway in Europe through the European and Mediterranean Plant Protection Organization. In 2012, EPA, in collaboration with Health Canada's Pest Management Regulatory Agency and California's Department of Pesticide Regulation, presented a white paper to the agency's scientific advisory panel describing a conceptual framework for assessing risks to bees. In 2014, based on the white paper and input from the scientific advisory panel and additional guidance documents that were generated through the European Food Safety Authority, EPA and Pest Management Regulatory Agency of Canada released a harmonized guidance for assessing pesticide risks to bees. This guidance has subsequently been translated into Spanish for consideration as a NAFTA harmonized guidance document. Slide 13. In addition to the 2014 guidance, in 2016, EPA issued an additional guidance document for agency risk assessors, which discusses the regulatory provisions for requiring data, the current data requirements for conventional pesticides, and additional bee toxicity and exposure studies. These studies include laboratory-based studies of individual bees and field-based studies of whole colonies, as well as residue monitoring studies in pollen and nectar. Slide 14. This is a simplified depiction of the three-tiered process used to assess risk to bees, where answers to multiple questions determine the extent to which higher-tier studies or refinements are needed. For example, the most basic question is whether it's likely that bees are exposed. This involves understanding whether the crop may be attractive to bees, whether applications occur at or near bloom, and the time at which the compounds are applied in terms of time of day. Also, transitioning to higher levels of refinement is influenced by whether mitigation measures may be available and the nature of data with which the risk assessments may be refined. As noted earlier, the process used to assess risk to bees is consistent with the process used with other taxa in that it's tiered. It consists of a screening level process, Tier 1, in which conservative estimates of exposure and effects to individual organisms, that is, adult or larval bees, are used to quantify potential risks. The screen focuses on what are considered to be the major routes of exposure, that being contact with spray or dust on foliage or in the air, and ingestion of residues in pollen and nectar. Higher tier assessments are conducted in which measured rather than modeled estimated exposure values may be used, and evaluations include colony level data. As with other taxa, the process relies largely on guideline or EPA or OECD studies, although one of the studies, residues in pollen and nectar, does not have a formal guideline or guidance. Also, as with other taxa, the process considers multiple lines of evidence, including registrant-submitted data, open literature, residue monitoring data, and incident data. Slide 15. In March 2016, the Government Accountability Office released a report to congressional requesters on its review of USDA and EPA efforts to protect bees. With respect to EPA, the Government Accountability Office reviewed the agency's efforts to protect bees through regulation of pesticides. GAO reviewed EPA's guidance for assessing risks to bees. It also interviewed agency officials and stakeholders from various groups, including beekeepers and pesticide manufacturing companies, and it published a report on additional actions that EPA should take to address threats to bee populations. In the recommendations, it indicated that the EPA administrator direct the Office of Pesticide Programs to develop a plan for obtaining data from pesticide registrants on the effects of pesticides on non-honeybee species. We refer to those as non-apis species, including other managed or wild native bees. Slide 16. Last year, EPA, to address these concerns, and as part of the process, EPA hosted a workshop on developing an exposure assessment paradigm for non-apis bees. The workshop was sponsored by the Pollinator Partnership and was organized similar to a Society of Environmental Toxicology and Chemistry, Pelston, in its efforts to have roughly equal representation from industry, government, and academia. The intent was to bring together a broad range of experts on non-apis bee biology, ecology, ecotoxicology, pollinator risk assessment, and there were a total of 40 participants, 35% from academia, about 40% from government, about 23% from industry, and one representative of a non-government organization, that being the Pollinator Partnership. Slide 17. Going into the workshop, there was general agreement among participants that the exposure pathways of honeybees are relatively well characterized. These include exposure of adult bees via contact and sprays and or residues on plant surfaces. Ingestion of residues in various forage items such as pollen and nectar or water or glutation fluid or propolis or honeydew also serves as a route of exposure. While there are multiple routes of exposure, contact and ingestion of residues in pollen and nectar are considered to be the predominant routes of exposure in the EPA risk assessment process for bees. While non-apis bees may share similar routes of exposure with honeybees, There are alternative routes of exposure through contact or ingestion with mud or leaf material, and there is uncertainty regarding the extent to which the alternative routes may represent a predominant route of exposure. Slide 18. The workshop focused on the extent to which honeybees are reasonable surrogates for modeling exposure to both apis and non-apis bees. The non-apis species considered in the workshop included managed social bees such as bumblebees, solitary bees such as mason bees, leafcutter bees, Megachile, and alkali bees, genus Nomia. And because Brazil was participating in the workshop, we also considered the stingless bees belonging to the tribe Meliponini. And the intent was of identifying current available science regarding the assessment of exposure to non-apis bees to pesticides to identify where additional research may be necessary. These species were selected because they're representative of social and solitary non-apis bees. They're also because they have economic value in providing pollination services, and since they're managed pollinator species, they would be readily available and can thrive under confined conditions. Slide 19. Discussions during the workshop focused on multiple routes of exposure for non-apis bees, and an effort was made to gauge their relative importance and whether they are or are not protective as those currently considered for honeybees. That is, contact exposure and ingestion of residues in pollen and nectar. Exposure equations for honeybees were discussed to identify the data which would be needed to develop similar algorithms for non-apis bees, with the goal of actually parameterizing these equations or to identify where additional research data are needed. Proceedings from the workshop have been submitted for publication in a peer-reviewed journal, and the target date for that publication is late this year. Slide 20. Some of the challenges identified in the workshop included the recognition that while methods are continuing to evolve for assessing both exposure and hazard to non-apis bees, resources have to be considered as to whether it's sufficient to implement these tests on a routine basis, or should a more strategic process be used. Also, in the European Union and elsewhere, there are efforts to reduce the extent of animal testing and place increased reliance on high throughput or in vitro assays, and or rely on safety factors to account for interspecies variability. Slide 21. In evaluating the potential risks to non-target organisms, there is a practical limit to the number of species that can be reasonably tested. While ideally we'd like to have data on all organisms and all possible routes of exposure, the sheer number of possible organisms and permutations of exposure routes in the environment vary widely. Therefore, the number of studies that are typically included have to be considered in terms of the cost, in terms of money and resources that are needed, such that they are not prohibitive. Slide 22. Honeybees have served as surrogates for terrestrial invertebrates for well over 20 years and have met the established criteria for use as surrogates. While non-apis bees have been identified as possible surrogates and represent managed native bees, current testing requirements for honeybees include 5 laboratory-based studies, and depending on the results of that screening level assessment, may include colony-level studies as well. Relative to other taxa, this is a large number of studies. While the studies are resource-intensive for the regulated community, the tests also require agency resources to review. However, available resources are becoming increasingly limited. Slide 23. So then, in response to the recommendations to the Government Accountability Office, the process for evaluating non-apis bees consists of EPA's continued reliance on honeybees as a surrogate for non-apis bees. Where data are available for non-apis bees, EPA will consider these data on a case-by-case basis to determine the extent to which honeybees are not suitable surrogates. Data on non-apis bees will continue to be used to qualitatively characterize potential hazard of non-apis organisms. At this time, though, available data do not support modifying data requirements for pollinators to include testing of non-apis bees. Slide 24. Another activity that is responsive to the national strategy is EPA's efforts to mitigate potential risk by restricting the use of pesticides that are toxic to bees. Slide 25. As part of the effort to restrict the use of pesticides that are toxic to bees, in 2017, EPA released its policy to mitigate the acute risk to bees from pesticide products. This policy included two mitigation measures, one which outlined label restrictions for use on specific products on crops where bees are brought in to provide contracted pollination services. The second mitigation measure highlighted state tribal managed pollinator protection plans to help limit exposure or risk to bees not under contract to provide pollination services. The acute risk mitigation policy prohibits the use of certain compounds while also providing flexibility for growers in some circumstances. Slide 26. The policy only applies to products meeting specific conditions. Those with foliar applications registered for use at bloom on pollinator-attractive crops where bees are present to provide contracted pollination services. Where these conditions are met, EPA has been working with registrants to ensure that the application restrictions are included on the label. Again, the label restrictions only apply when the target crop is in bloom, a contract exists for pollination services to the target crop, and managed bees are present to provide pollination services to the target crop. Slide 27. This is the actual label language. However, as I indicated, there is flexibility for growers. This flexibility allows for applications at night for products with residual toxicity times of 6 hours or less, and for applications for crops with indeterminate blooming periods. Applications can also be made to indeterminate blooming crops when air temperatures are 50 degrees Fahrenheit or less. Slide 28. One of the conditions influencing the acute risk mitigation restrictions is the toxicity on residues on foliage time. This is referred to as the RT25. The RT25 is intended to be a measure of the time that a pesticide product is expected to remain toxic, or more specifically, it's the time that a product remains toxic to less than 25% of the bees exposed to weathered residues. The study is conditionally required for products with an acute contact toxicity of less than 11 micrograms per bee. The RT25 has historically been considered by both beekeepers and growers to be a useful metric for indicating which products may be less toxic to bees when applied at bloom. In response to requests from EPA's Federal Advisory Committee, the Pesticide Program Dialogue Committee, the agency has posted the RT25 data to its website. Slide 30. In reviewing the RT25 data prior to posting on the web, a cursory analysis suggested that there is considerable variability in the chemical physical characteristics of the pesticide's active ingredients and their RT25 values. Also, there is uncertainty regarding the extent to which the RT25 values are predictive for different formulations of an active substance under actual field conditions and how weathering may affect these values. Slide 31. To address these uncertainties, EPA has been coordinating with the Pollinator Research Task Force. Its acronym is the PRTF. The PRTF is a consortium of technical staff from the regulated community, and the goal of this group is to generate data that may help to inform the pollinator risk assessment process and to coordinate and collaborate with EPA regarding pollinator data requirements needed to support the registration or the re-registration of pesticides under the Federal Insecticide, Fungicide, and Rodenticide Act. Based on slide 32. Based on discussions between EPA and the PRTF, the initial focus of research on the RT25 is to better understand or to standardize parameters included in the toxicity residue on foliage guidance document. It's guideline 850-3030. In the initial phase of testing, proposed modifications will be tested by contract research laboratories in 2018. Longer-term improvements in the guidance may include identifying suitable reference toxicants or positive controls that can be run in the study, and determining the extent to which formulation and weathering conditions may influence test results. Slide 33. The national strategy, as well as the acute risk mitigation policy, identified opportunities for mitigating risks of pesticides to bees through the development of state or tribal managed pollinator protection plans. Slide 34. In general, EPA is promoting managed pollinator protection plans, abbreviated MP3s, as a means of mitigating exposure from acutely toxic pesticides for managed bees not under contract for pollination services. Roughly 48 states have MP3s in some stage of development, and at least 10 tribes have pollinator protection plans that focus on native pollinators. States and tribes are not required to develop the plans, and they have the option of adopting a regulatory approach or a voluntary approach. The scope of the plans can be expanded to address other pesticide-related issues and can be expanded to include other factors impacting pollinator health. While EPA is reviewing the state or tribal MP3s, it's not approving these plans. However, EPA has encouraged the states and tribes to develop and implement the plans as quickly as possible. Slide 35. EPA is continuing to work with states and tribes on developing a means of evaluating the efficacy of MP3s. As part of this effort, in 2016, EPA formed a work group with the Pesticide Program Dialogue Committee to provide recommendations on various metrics which could be used in evaluating MP3s and how to communicate their effectiveness to the public. Slide 36. As a result, the PPDC recommended the development of a survey instrument that state lead agencies have taken the lead on in development and deployment of the survey. The survey to states focuses on elements of the MP3 that have been common among the plans that have been generated thus far, and they include measures of communication, development of best management practices, and standard operating procedures to reduce exposure of pesticides to bees. Also, efforts towards education and outreach and engagement with stakeholders. Also, examining measures of behavioral changes. Slide 37. Another of the commitments which EPA made in the national strategy was expediting the reviews of products used to control varroa mites. As indicated early in this presentation, pests are one of the factors influencing declines in honeybee health, and varroa mites and the viruses which they vector have been seen as serious challenges for beekeepers. Slide 38. There are a variety of products currently available to beekeepers. However, the efficacy of some of these products has waned due to the development of mite resistance. In 2013, EPA registered the use of the miticide amitraz for use in bee colonies, and in 2015, EPA registered the use of oxalic acid, where USDA is serving as the technical registrant. Slide 39. EPA and USDA have been working with the Honeybee Health Coalition and a consortium of researchers in the U.S. and abroad to identify additional products that may be effective varroacides. These researchers are not only looking at whether products may be effective, but they're also examining how resistance develops among mites. The research is being funded through a combination of monies provided by a grant from the Foundation for Food and Agricultural Research and through contributions from universities and private organizations. Slide 40. So I've provided an overview of some of the things which EPA is engaged with respect to assessing and managing risks to insect pollinators. As I've indicated, there are multiple factors associated with declines in bee health, and EPA is focused on ensuring that the best science is brought to bear on assessing and managing risks to bees from pesticides. Again, I want to thank you for the opportunity to talk about some of the things which EPA is engaged in to address the declines in pollinator health.
Great. Thank you very much, Tom. At this moment in time, we will open the stage for any questions. And I will lead us off with the first question. Tom, the first question we have is, to what extent has the research identified in the Pollinator Research Action Plan of the National Strategy been addressed?
Could you repeat that again, please?
Sure, no problem. To what extent has the research identified in the Pollinator Research Action Plan of the National Strategy been addressed?
the Pollinator Research Action Plan had a number of research actions across all of the different factors that are known to affect pollinator health. And each of the agencies that identified those different research items has the task of tracking the different elements that they identified. So relative to pesticides, I've discussed the efforts that EPA has done, but the other research efforts in terms of looking at varroa mite or pests and controlling diseases, each of those is being tracked separately by their respective agencies. And each year a Excel spreadsheet is provided to the White House on where those activities are at.
Great, thank you. One other question that we have received is, are agrochemical companies the only source of information the EPA considers in assessing potential risk of pesticides to bees?
The agrochemical companies, or the registrants, are required by law to generate data to support the registration of pesticides. The public is not expected to finance the generation of those data. With that said, though, the agency relies on multiple lines of evidence, including open literature, monitoring data, and incident data, where bee kill reports have been submitted, to inform our risk assessment process. And where open literature studies are considered, those studies are held to the same standard that the regulated community is held to, and the guidelines that the EPA uses to assess open literature studies is posted to our website.
Fantastic. I think that is our wraps up our questions for Dr. Steeger. Thank you again for taking the time to present with us. And now we will move forward to Dr. Troy Anderson, who will be the second speaker of today's ACS webinar. All right, Dr. Anderson is Associate Professor of Insect Physiology at the University of Nebraska-Lincoln, where his research program studies the broader aspects of physiology and biochemistry that involve the integration of toxicological, pharmacological, molecular, and genomic approaches to address fundamental questions in biology. Today, he's going to be talking about pollinator protection activities. Troy?
All right, thank you, Daniel. Good morning or afternoon, wherever you might be listening today. I want to say first, it's a pleasure to share this opportunity with Tom Steeger, who just gave a wonderful presentation on what the EPA is doing with these pollinator protection activities. I also want to thank John Clark for the invitation, Claire Terry for organizing this webinar, as well as Daniel Swell for moderating it, and also Kyle for his technical assistance to make sure we get online properly. As Daniel mentioned, our contribution to pollinator protection activities is related to more integrative, physiological, biochemical, toxicological approaches to study pollinators, pesticides, and parasites. Let's see if I can continue on here. There we go. So as Tom had mentioned, beekeepers are continuing to report high annual losses, with average totals in the country being around 35% as it stands. The states that I've worked in, Virginia and Nebraska, it's a bit variable, where we have anywhere from 30 to 70% losses between those two states. And there are multiple stressors that interact with each other, as Tom mentioned, that can negatively affect bee health. This can be malnutrition, it can be parasite infestations, disease infections, and really what we focus on are these bottom two here, being pesticides and the pesticides that are used for parasites within honeybee colonies. So our overall goal is to characterize the risk of these pesticides to honeybees, and we do this by measuring the fate, persistence, and application of pesticide exposures in a variety of different environmental matrices, whether it's seed treatments, surface waters, soils, plants, and how they move from these matrices from foraging workers back to the hive, and then are distributed through the different hive matrices, whether that's within wax, pollen, the bees themselves. We conduct a variety of different laboratory field toxicity testing, bioassays, again with our overall goal in determining risk or estimating risk, predicting the effects of these pesticide use, misuse, and their safety to these pollinators. The purpose of this and why we do this is because it's a key requirement to provide safety to bees for these insecticide exposures, especially when you're considering the development and re-registration of insecticidal chemicals. We also do this to develop a better understanding of the biochemical and physiological interactions that these insecticides have with honeybees and other bee pollinators. We also want to identify natural mechanisms of selectivity that provide resistance, or I should say tolerance, to insecticides in bee pollinators, and then also identify bee pollinator-friendly chemical scaffolds for rational design. But I think one of the most important points here, and as Tom alluded to, is that we want to improve science-based communication with regulatory authorities about the risks that are posed by commercial insecticides. So what do we know about bee sensitivities to these insecticides? Well, in 2006, Claudianos et al. Published the honeybee genome, which annotated 11,000 genes that showed marked differences between cytochrome P450 monooxygenases, general esterases, and glutathione S transferases. So all detoxification genes that are important for metabolizing insecticides. And what they found is that there are a lower number of these genes compared to other insect species. But what they report in this study is that this deficit of detoxification genes in the honeybee will translate to less pesticide detoxification capability, which may explain the species' unusual sensitivity to pesticides. Interestingly, Melissa Hardstone and Jeff Scott provided a paper in a study in 2010 where they reported that honeybees were no more sensitive than other insect species across 60+ insecticide classes. This included carbamates, neonicotinoids, organochlorines, organophosphates, and pyrethroids, as well as other miscellaneous insecticides. And if you look through the literature, you find these differences in selectivity between bees and these chemistries and pest species. For example, with the neonicotinoids that make up about 20% of the market share that target nicotinic acetylcholine receptors in insects, you find that with thiacloprid, the LD50 for this compound is around 39 micrograms per bee, whereas with the green peach aphid, it's about 0.39. So you see this 100-fold difference between selectivity for this compound. If you look at imidacloprid, you see about a 5-fold difference between honeybee and green peach aphid. But what's interesting, if you look at the two different compounds for each species, there's about a 4-fold difference in its toxicity between thiacloprid and imidacloprid to the green peach aphid, and over a 2,000-fold difference in selectivity between these two neonicotinoids with the honeybee. We also see similar results with pyrethroids. So talfovalinate, which is an in-hive acaricide for varroa mite control, has an LD50 around 10 micrograms per bee, but with cotton bollworm has about a 1 nanogram LD50 per larvae. So again, about an 8,000-fold difference here. But if you look at delta methrin, it's only about a 5-fold difference between the two species. If you compare the two pyrethroids with each other, there's almost a 7,000-fold difference between fluvalinate and delta methrin to honeybees, and about a 4-fold difference between the two chemistries with the cotton bollworm. We see similar selectivity issues with in-hive acaricides that are used to treat varroa mites within honeybee colonies. So talfovalinate is a pyrethroid class acaricide that has an LD50 around 2 micromolar, and an LD50 of 9 micromolar for varroa destructor. So about a 4.5-fold difference. Again, a marginal difference in the selectivity with coumaphos between these these two arthropods. With amitraz, we see about a 50-fold difference in that this compound is more selected towards varroa mite compared to honeybee. So there's many reasons for why these differences in selectivity exist. One of those could be reduced cuticular penetration based on the physiochemical properties of the insecticidal chemistry. It could be increased transcription, gene duplication events that basically elevate the level of detoxification proteins to metabolize these compounds. You can have increased target site insensitivity with mutations in say, a voltage-gated sodium channel or acetylcholinesterase in many cases. And a relatively new player to this game are the ABC transporters, which basically act as molecular vacuum cleaners to increase cellular efflux of these insecticides across the blood-brain barrier. So one of our studies that we conducted was to examine the level of pesticide residues in beehive matrices. This is a follow-up study to a great project that was conducted by Chris Mullen's group at Penn State, where they looked at 120 different toxicants within honeybee colony matrices in North America. And what was interesting in the study is that these pesticides occur as two or more mixtures and not just one chemistry itself. And if you look through the data, you find that 99% of the bee colony matrices, whether that's wax, pollen, or bees, have pyrethroids and organophosphates. 60% of those matrices contain fungicides, about 50% contain herbicides, 20% carbamates, and only about 3% contain neonicotinoids. And so they go on to discuss what the implications of these pesticide residue exposures may mean to a honeybee colony health. So with our study, we focused on 50-plus apiaries in Virginia, where, again, we tested pesticide residues in bees, wax, and pollen, working with beekeepers across the state to be able to do this sampling project. What we found were a variety of different pyrethroids, organophosphates, triazine herbicide, atrazine, and the fungicide chlorthalonil. What I want to draw your attention to is that the bulk of our samples here contain the N-hive acaricide fluvalinate, with the highest detections being in the wax. Next, we find the N-hive acaricide coumaphos and its O-analog metabolite coralox. Again, maximum detections found in the wax and pollen. And lastly, we found quite high levels of chlorthalonil within these hives too, a broad spectrum fungicide that is used in cropping systems. So these acaricides, as Tom had mentioned before, are used to control this major apicultural challenge, Varroa destructor, or the varroa mite. So this varroa mite that you can see on the thorax of this bee here is a hematophagous mite. It feeds on the hemolymph of bees, and it transmits multiple viruses that affect the physiology and overall health of that bee. And so what are available to beekeepers are a variety of in-hive acaricide chemistries, one being talfluvalinate, which is a pyrethroid class acaricide that targets voltage-gated sodium channels. It was registered in 1988 for use to control varroa mites. In a sense, there have been issues with knockdown resistance or KDR resistance associated with this compound in varroa mite populations, which lead to its low efficacy as an acaricide. Another one would be coumaphos, which is an organophosphate class acaricide that targets acetylcholinesterase. It was approved in 1999 for varroa mite control, and since there are reports of metabolic resistance associated with this compound, again making it a low efficacy acaricide. The third one I want to mention today is the formamidine amitraz, which targets octopamine receptors in the mites, and it was approved in 2010. And since then, still no reported resistance issues associated with this compound. It does provide high efficacy for controlling varroa mite populations. For those of you that are not aware of these applications, basically these acaricides are provided on strips and put into the colony where the bees walk on the acaricide strips. They pick up the molecules and distribute them with each other via contact, and then mites that are exposed to these acaricides can either be repelled or paralyzed and removed from the bee. So our questions were, how do these in-hive acaricides affect bee health? So we set up a field study where we treated colonies with apistan, which contains talfluvalinate at 10%, and checkmite, which contains coumaphos at 10.3% for a 6-week period according to manufacturer recommendations. We also measured, in any of these studies, we measured total protein, total carbohydrate, and total lipids in bees after the 6-week exposure period. And what we find is that both total protein and carbohydrate levels are significantly reduced in these acaricide-treated bees. However, there were no differences in lipid contents in the bees. We also wanted to identify what the effects of these acaricides might be on individual and group level immune parameters. And so on the individual side, we looked at phenol oxidase as a marker. And again, what we show is that these two acaricides significantly reduce phenol oxidase activity in the treated bees. And as a measure of group immunity, we use glucose oxidase. And we show that there's a significantly higher level of glucose oxidase activity in bees that are treated with these two acaricides compared to the untreated colonies. Next, we examined the gut microbes within these acaricide-treated bees. And what we find is that the bee gut microbiome is dominated by actinobacteria, firmicutes, and proteobacteria, as well as fungal communities of Ascomycetes and Basidiomycetes. What's important about the study that we find is that the acaricides, fluvalinate, and coumaphos significantly change the structure and function potential of this gut bacteria community. Not only these two acaricides, but also that fungicide chlorthalonil has an effect on the gut microbes within the bees. We've also done some work with amitraz, where we have shown that amitraz not only affects the cardiac activity of bees, but it also decreases survival in virus-infected bees. And so in the bottom panel here, the panel graph, I should say, we exposed bees to amitraz or its metabolite DPMF, and also octopamine, which is a known cardioaccelerant. And then we use phentolamine as a blocker of octopamine receptors to measure this cardiac activity. And again, what we find here is that with amitraz and DPMF, you get a significant increase in cardiac activity, or the beats per minute of the bee heart compared to the untreated bees. And if you use phentolamine as a blocker, you can block that effect, so confirming that these do have an effect on cardiac activity. In the top two graphs, you see that bees that are treated with amitraz and infected with the virus, there's a decreased survival of those infected bees compared to the amitraz untreated bees that are infected with the virus. Similar effect with DPMF, or that metabolite of amitraz. So one of our major challenges in the apiculture industry is the delivery of acaricides. And these issues surrounding acaricide delivery, they kind of go back to the same mechanisms that I discussed with bee selectivity, in that you can have physiochemical properties that reduce cuticle penetration, you can have differences in detoxification, enzyme activities, you can have target site insensitivities, and then also ABC transporter efflux issues. And so what we have found is that toxicokinetics does limit acaricide delivery to the intended target site, or the movement of these acaricides to their target site. And so in this study, we measured the field efficacy of apistan, checkmite, and apivar, so talfluvalinate, coumaphos, and amitraz. And in 28 colonies that we tested, we found that there were 5 varroa mite populations that were resistant to talfluvalinate, and out of 38 colonies, we found that there were 15 varroa mite populations that were resistant to checkmite. Of 16 colonies tested, we didn't find any that were resistant to amitraz or apivar. But in these resistant varroa mite populations, we find that there are significant increases in general esterases, glutathione S transferases, and cytochrome P450 monooxygenases. We also know that toxicodynamics limit acaricide activity or the interaction of the acaricide chemistry with its intended target site. So there's been some exciting work from Joel Gonzalez Cabrera on identifying novel mutations within sodium channels of pyrethroid-resistant varroa mite populations, where they're finding, mutations within the selectivity filter of the sodium channel at domain 4. There are also studies that are showing some issues with anticholinesterase compound binding between acetylcholinesterase of honeybee and varroa mites. And what these authors report is that there's this phenylalanine 386 to leucine 347 difference between the active site and peripheral site of acetylcholinesterase that has an effect on the sensitivity of organophosphates or carbamates to these two acetylcholinesterases, making them less potent to honeybees. And we have shown in our work with resistant varroa mite populations that if you look at residual acetylcholinesterase activity when exposed to coumaphos-oxon or the O-analog metabolite of coumaphos, you see a substantial difference in the potency between the resistant and the susceptible varroa mite populations. So basically demonstrating that there may be some sort of target site and sensitivity issue there, although we're not aware of what that mutation might be that confers this difference in selectivity. So, which leads us to our current work with still-being chemistries as alternative chemical interventions for varroa mites. And the purpose of using these still-being chemistries is because they provide a new mode of action for varroa mites and also a way to mitigate resistance issues that are associated with the currently used acaricides for varroa mite control. So we chose these still-beans based off of a natural product, stilbenoid scaffold, and in the lower left hand you see is this dihydroxy-4-isopropyl stilbene compound that was isolated from Photorhabdus bacteria in nematodes. And so basically what happens here is that you have the nematode that infects an insect host, it releases this toxicant to kill the insect host, as well as antibiotics to fight off any microbial competitors. The bacterium will colonize the nematodes and the process starts over again. So this has been work in collaboration with Jeff Bloomquist at the University of Florida, where he has shown that these stilbenes have nematocidal activity and have an effect on anion transport, blocking anion transport in different nematodes. We've done follow-up studies where we show that these stilbenes have effects on voltage-sensitive chloride channels, and up in the top right-hand corner in panel B, you can see that there is this dose concentration response effect here that inhibits these chloride channels within SWA1B cells of mosquitoes. But what about the selectivity or the toxicity to bees and varroa mites? Well, we see quite large differences here in their effects on these two species, in that with DIDs we find an LD50 right around 10 millimolar, which is about the same for all three of these compounds, but with varroa mites about 150 micromolar, or a 65-fold difference. We see about a 5,000-fold difference between 2-methoxystilbene, a 70-fold difference with 3,5-dimethoxystilbene, and only about a 2-fold difference with the methoxysterophene. So our next question was, how do these products, these chemistries work in the field? So here, to walk you through these field efficacy bioassays, as a former student of mine, Feline Vu, takes a frame from the hives, collects mites from the brood frames of each bee colony, and she collects about 300 brood nest bees, and she exposes them to acaricide-treated tabs for 3 and 6 hour intervals. And so in this panel here, you can see this container. In the middle there is a tab with a acaricide strip tab that's hanging here. There are about 300 bees in the container. In between these two wood platforms is a screen, and at the bottom is a sticky board to collect any mites that drop from the bees. After the 3 and 6 hour exposure periods, we wash the bees with ethanol to collect any remaining bees or mites that may be on the bees. So again, on the bottom here, you see the number of mites for the 3 and 6 hour drop, the number of mites that were recovered in the ethanol wash, the total number of mites, the efficacy based on these numbers, the total number of bees, and then the total number of mites per 100 bees. So anything that showed below 60% efficacy, we consider that as a resistant varroa mite population. And if we had 5 or more mites per 100 bees, we considered it a valid test. So these are our data. Again, in the first graph here, we show that out of 28 bee colonies tested, we found 5 varroa mite populations that were resistant to apistan or talphovalonate. And out of 38 bee colonies, we found 15 varroa mite populations that were resistant to checkmite or coumaphos. And again, no resistance found with apivar in these bee colonies or to the varroa mites. So how do these stilbenes compare to these current acaricides where resistance has been documented with the varroa mite populations? So basically what we have here in this graph are, on the x-axis we have DIDs, we have 2-methoxystilbene, 3,5-dimethoxystilbene, and then the 4-methoxysterols. And in the left side here, these are bees that were treated with apistan strips, or mites treated with apistan strips, showing less than 60% efficacy, or what we consider resistant to this chemistry. And with these 3 stilbenes, we don't show comparable efficacy, actually reduced efficacy, compared to apistan. But what's significant here are the mites that are treated with DIDs. And we see a significant increase in efficacy compared to the apistan treated mites. And we see a similar effect here with the check mite resistant mites. Again, these 3 stilbenes do not have a comparable effect or have lower efficacy than checkmite itself, but DIDs significantly increases the efficacy, has significantly increased efficacy compared to checkmite in these populations. So just to finish up here, we know that bee decline is a nationally recognized problem, demands attention from both the scientific community and beekeeping industry, and the acaricide use is one of the primary perceived problems for the bee decline. We found that nutrition immune responses are observed in bees that are exposed to acaricide treatments, and it's good to understand that this malnutrition event can weaken immune responses to parasite infestations and pathogen infections. We also found that acaricide treatments can alter the microbiome thresholds in the colony, and so disrupting bacterial and fungal communities that are important for nutrition and immune health of bees. We know that widespread acaricide resistance does limit the use of standard in-hive chemistries to reduce varroa mite infestations, as well as the diseases that they transmit. And we're continuing to work with several of these stilbene chemistries to provide alternative chemical interventions to be able to either deplete or incapacitate varroa mite populations, and again, in turn, reduce infectious diseases in bee colonies. And Tom had mentioned this before, is that we're part of the group with the Honeybee Health Coalition to provide new acaricides for varroa mite management. And lastly, I just want to recognize those that are involved with these projects and upcoming work. My group here at University of Nebraska, Jen Williams, Bryant Gabriel, Ellis Johnson, and Drs. O'Neill and Ralt. Also the group that I work with at Virginia Tech, in particular Dr. Rick Fell and Dr. Carlisle Brewster. And then my collaborators at University of Florida and Louisiana State University, again, Drs. Bloomquist and Swell. And with that, I will wrap up and be happy to take any questions that the audience might have.
Great, thank you very much, Troy. The first question we have is, your stilbene data is very interesting and exciting. Are there any new structural classes of anticholinergics that are promising candidates for varroa control that might be able to take advantage of the differences between the mite and bee protein chemistry or protein biology?
There are. We have been working with a series of anticholinesterases based off of data that we have collected with carbamates, standard carbamates. We show anywhere from 100 to 500-fold differences in the potency of these carbamates, being more selective for varroa mite acetylcholinesterase compared to honeybee. So we're working with Jeff Bloomquist right now, and Paul Carlier, one from Florida, one from Virginia Tech, to look at experimental carbamates to see if we can tease out what these differences are between the two acetylcholinesterases.
Great. The next question is, what are your thoughts on hygienic bee strains, and does the preliminary success of these strains reduce the importance of novel chemical development for Varroa control?
Yeah, that's a good question. We haven't worked with any of these acaricides and hygienic strains. I'm not sure, how that's going to have an effect. Definitely, if the hygienic strains are managing lower thresholds of varroa mite populations within the colony, then you definitely don't want to seek out chemical treatments for those. So I think as long as those are maintaining themselves and below thresholds or keeping varroa mite populations down to a manageable level, then I think the chemistries are not exactly necessary at that point. I think the chemistries right now, new modes of action are needed because the current ones we have, there's just a variety of different resistance issues associated with them. So I think any new mode of action that you develop and that you test with varroa mite populations are going to show considerable results compared to the current use chemistries.
Fantastic. The last question that we have is, how do drought conditions affect bees, and will they affect any of these miticides that you presented on?
Was that drought conditions?
Drought conditions, correct.
Yeah, so the easy answer is I'm not sure how drought conditions will affect the use of these acaricides. We have, basically because of our field efficacy testing, we collect bees from the hive and test them with the tabs rather than putting full strips into the hive to see what their, residual effects are going to be, the persistence is going to be, and whatnot. So I really don't have any field data or information on knowing how those will affect. With their stability, that needs to be tested as well. So there are a lot of formulation issues that need to be considered before these can actually make it to commercial use. So,
great. Well, those are all the questions that we have. So thank you very much, Dr. Anderson, for taking the time to present today in ACS webinar.
Thank you.